So, what if the assumptions to the t-test are violated? Now, this isn't a time to panic. Um, Hopefully, you have done everything you can to make sure that the quality of your data is all in order. So, sometimes we have um, non-normal distribution if one of our data points is looks like it's an outlier. Perhaps that one was typed in wrong. So definitely go back and double check the correctness of all your data. Um, if possible, you can redo the experiment. So it depends on how big your experiment is, how long it's taken you, how much money you have. You might be able to redo it with a larger sample size or additional measures to control variance. All right, so better job controlling or your preliminary data, you may discover there's different kinds of individuals in there. And if you separate them out, then your result is going to look a lot cleaner. Hopefully, you did that already in a pilot study, which would be sort of a mini version of your experiment just to see how things went before you actually did the whole complete experiment, right? Depends on how big it is, how complicated, how long it's going to take. Right. Another thing I forgot to put on the slide is that sometimes you can do what's called a transformation on your data. Certain kinds of um, deviations from the normal distribution can be corrected. And mathematically, you can do the statistical test on the transformed data. So taking the log, taking the square root, depending on what shape your distribution is, those things can um, uh, fix the problem of your data not being normally distributed. All right, so transform the data is another option that's on this slide. But if none of those are an option, then what we're going to talk about today is these non-parametric. Sometimes people call them distribution-free, which has a more positive connotation. So I like that one as well. You're not worrying about being tied to this distribution. But generally, they're called non-parametric tests. Well, why are they called non-parametric tests? Because the typical tests that we do, t-tests, for example, are parametric. Remember how we called the mean and standard deviation of the population parameters, and we represented those as mu and sigma. And we called the measurements we took on our actual sample, the sample mean and standard deviation, we called those statistics. All right, so some distribution where we have parameters that are going to determine it, that's where the term parametric comes from. And so we're using that distribution, and we are assuming that the parameters are matching our data. And for this class, basically, we're going to be using the normal distribution as the distribution. So in a parametric test, we're going to uh, use the test statistic calculated based on the assumption of the normal distribution to find the p-value. Now, these tests are usually the ones we want to do if we can. They're more powerful. That means it's easier to reject the null hypothesis, and we'll see in a few minutes when I show you an example of how to do a non-parametric test. We're actually chucking out some of the information. Right, so the parametric test is basically going to use every observation, the actual value of it, whereas the non-parametric test is only going to look and see what order the data go in. All right, so non-parametric tests are not going to use a model distribution, and we have to change what we're actually doing when we do the test. So this is really important. They test hypotheses about medians instead of means. All right, so we're not going to assume it has a normal distribution. The mean isn't going to help us out. All right, so it's possible that we have a test, some data that we want to test. We want to do a test for the median for some reason because if we know how the data is distributed, and that is our variable of interest. And so we could go straight to one of these tests if that were the case. All right, so they can also test data that is not quantitative, because we need to just put them in an order or rank them. And so data that are scales or some kind of um, qu- calculated quantity, some kind of metric, we can do non-parametric tests on those kinds of data that isn't strictly quantitative, and any kind of information that sort of we've put things into two categories. So I'm putting on here binomial, right? So that either a yes or a no. We can have the results of a survey where people liked or didn't like the movie, and we can see if significantly more people liked the movie than didn't like the movie. All right, so we can use especially one of the tests, the sign test. We can use it on all kinds of kooky situations that we may not uh, have the kind of data that you normally think about that we're going to be doing these tests on quantitative variables, right? So if you have some wacky data, you have absolutely no idea what to do with it. Um, yeah, think of the sign test. Okay, so the tests are less powerful. That means it's harder to reject the null than the equivalent parametric test. All right, so we need to be seeing an effect that is pretty strong if we're going to be able to find a significant result using these types of tests. All right, so let's review the assumptions for our t-tests because we're going to talk about the two kinds of tests that are the equivalent to the paired t-test and the two independent samples t-test. All right, so if we want to do a t-test, we need to have the data normally distributed or close to normally distributed. Um, or they have a large enough sample size, right? So our data don't have to be perfectly normally distributed. If for the one sample or paired test, our sample size is over 15, or for our two independent samples test, our two sample sizes are equal and they're greater than 5. Now, if they're unequal, they have to be even bigger, all right? But it's kind of hard to do an experiment that's meaningful, that doesn't meet these qualifications. However, two independent samples also has a assumption that the standard deviations are equal or approximately equal. Right. So if the standard deviations are very different between your two samples, then that could be an indication that you need to do a non-parametric test. Okay. So if we're planning on doing t-tests, we got to check the assumptions of the test and decide if it's going to be valid for the data that we have. If we decide that it's not valid, then we're going to decide on which test we're going to do. So this is um, an inset is or a little piece of our which test flowchart, and it's showing the branch that goes from differences between two means. And remember that the first branch you look at is whether the samples are related or they're independent, right? So just like we did with our t-test, that's still the first thing we want to think about when we're thinking about what non-parametric tests to use, right? So if we have related samples, if our assumptions met and we can do the metric version of the paired t-test, then if we need to do the non-parametric test instead, then we're going to use the sign test. So you can think of the sign test as the non-parametric equivalent of paired T, we're going to use it when we have related samples where we've measured the same control twice. We can use the sign test for other situations as well. So it goes beyond just being the equivalent of the paired t-test. If we're on the independent side, we have two independent samples. The parametric test is the two independent samples t-test. The non-parametric test, we're going to do the rank sum test. So you should think of it as the non-parametric equivalent to the same experimental design that we have when we're doing the two independent samples t-test. If you're using computer software or you go on to read more about non-parametric tests, you probably aren't going to read about the rank sum test. You're going to use a little more sophisticated test, either the Wilcoxon or the Mann-Whitney U test. All right, they're a little harder to calculate test statistic by hand. And so we're just going to do the rank sum test in class just because of the ease of doing it. We don't have a software package we're using that is going to do the other tests. However, when you go on in life and you're using a sophisticated software package, basically the same, uh, you'll use it in the same way. Just get a p-value from the software instead of having to work out all the steps by hand. All right, so I'm going to start with the sign test. And I'm going to explain to you how to do this test as we work through an example problem. All right, so we're going to have a set of data that we've decided we're going to do the sign test on. And this example asks the question, are feral cats more or less aggressive when they have kittens? Um, so brave volunteers went out into the neighborhood where these cats are living, and they did an interaction with them and used an aggressiveness score that's calculated based on behaviors. So this score is on a scale of 1 to 10, but it's this metric that's sort of calculated based on observation of some different things. And so we don't often things like that are calculated, some kind of metric or index is not going to have a normal distribution. All right, so we measured um, the cats during some part of the year that they didn't have kittens. And then we came back again during the part of the year where they do have kittens, and we checked where they actually had kittens. All right, so we've got the same 7, and we have a measurement for them, their score without kittens and their score with kittens. All right, so we need to have all of these data, these two measurements on the same paths. So this is the same way the data would look for a paired test. All right, now when we write the hypotheses for our sign test, they're going to be that the medians are equal for the null hypothesis, and the alternate is going to be that the medians are not equal, or we can have a one-sided alternate hypothesis as well. And what we're going to do these tests, our test statistic, is we're we don't actually have to take the difference of the measurements like we did. We're just going to look at the sign of the measurements, right? So this is called the sign test because we're really only looking at the relationship between the two numbers, right? So I've chosen in this example that looking at the data, most of these scores with kittens are larger, and so I subtracted the other value from it. Doesn't matter which way you do it. You could have done it across just saying, okay, I'm going to do 3 minus 7, I'm going to get a negative number. As long as you do it the same way all the way down, you'll get the same result, right? So I just chose to subtract 3 from 7, and I got 4, and that's a positive 4. So I did all the subtractions and I wrote down whether I got a positive or a negative number. All right, so these signs are in red because that's the only thing we care about. You don't even actually have to do the subtraction. So when I say when we're doing a non-parametric test, we're throwing out information or we're not using all the information. That's what I mean. Instead of seeing this big difference here versus this small difference of 8 versus 1, all we care about is it positive or negative. So whether it's a big difference or a small difference, we're not um, we're not. Okay. So we're going to call little s for the sign test. We're going to count up the number of pluses that we have. We're going to count up the number of minuses that we have. If there's a difference of 0, all right, suppose this measurement was 6 and then 6 again. All right, so there's going to be a 0 difference. We're not going to count it. So it's neither a plus nor a minus, right? Now, if we think about how this test works, if all of the differences are 0, then there's no difference between the values in the two treatments. All right, the median of one of those, the before measurements, is going to equal the median of the after measurements. All right, so if we have approximately equal medians, it's likely due to sampling variability that we'll have some pluses and some minuses, but the number will be equal to one another. Okay. So the more different the number of plus values and the number of minus values is, the more likely um, our medians all right. So we're going to look for a big difference between the number of pluses and minuses to consider our result significant. All right. So in this case, let's look here. We've got 7 measurements. We've got 1 minus. That means we have 6 pluses. So those are our results. And the way we're going to figure out the p-value is using the binomial distribution, but just for fun, we have a quick kind of cheat sheet we can use to test to see if we're going to accept or reject our hypothesis at the 0.05 level. All right, so this table was created at the 0.05 level, so it's only going to work if that's our alpha level. All right, and this is a zoomed in version of the full page sign test table you have in your table packet, and it has minuses starting at 1 going across the bottom, and the vertical axis is pluses, right? Now this middle region, this middle wedge is going to be the accept null hypothesis region. Right. And the reason that's in the middle is because each of these little squares is going to represent how many pluses and minuses you got. So if I'm here in the 1 and 1 square, we had 1 plus and 1 minus. In that case, the medians are equal. All right. So this 1-to-1 line that goes up the middle of the diagram is if our null hypothesis is true. All right. The farther we get from a equal ratio between our pluses and minuses, then the stronger the evidence is against the null hypothesis. All right. So this is symmetrical. So you can either have 6 minuses and 1 plus, or you can have 6 pluses and 1 minus. You're going to reach the same conclusion. And that is outside of these darker lines, that is the region of this diagram where you can reject the null. All right, so you can see from this that we tallied up for our test of feral cats that we had indeed 6 pluses and 1 minus. So we are in this reject the null hypothesis region. All right. So that's kind of a quick and easy way that we can evaluate the results of our sign test. Um, but look at how different the number of pluses and minuses has to be in order for us to reject the null, right? Not 5 and 2. It's got to be 6 and 1. Okay, so as good statisticians, we always want to report the p-value though. So for the sign test, we're going to use the binomial distribution to find an exact p-value for our example. All right, so we're going to use the binomial distribution. And the reason for that is that we have two different things that are happening when we're in the binomial setting. And according to our null hypothesis, we should have a 50-50 ratio of pluses and minuses. If the medians are equal, then we should have the same number of pluses and minuses if they're not all zeros. Okay, so we can use the binomial distribution. Remember that it has two parameters, little n and little p. So for our little n, we're going to use the sample size, but we're not going to include any values where the two measurements are exactly equal. All right, so if we have any values of 0 that are neither pluses or minuses, we're going to ignore them. All right, so this case, our total sample size is going to be 7, the 6 plus 1, the pluses and the minuses. And to find the p-value for the sign test, we're always going to use our binomial distribution with 0.5, all right, because that is what the null hypothesis situation is setting up for us. Okay, so this can get really confusing because we've got this little p. All right, that's the probability of the success that we're going to use because it matches our null hypothesis. All right, we have to use that distribution to find our p-value. Okay, and if our number of pluses and minuses are different, all right, we want to know the probability of getting an outcome as extreme or more extreme than the observed outcome due to chance if the null hypothesis is true. All right, so in order to find the p-value, we're going to look at the whole distribution. All right, so remember that the binomial distribution table, we have to find the chunk in the table for our sample size, which is 7. And there's a probability of getting 0 successes out of 7, 1, 2, 3, 4, 5, 6, or 7 successes out of 7. All right. So to find the p-value for the sign test using the binomial distribution, we're going to find the sum of the probability that x, our value in the distribution, is less than s, right? So we're going to choose the smaller of our s of the pluses and our s of the minuses. All right, and that's so that I can give you a rule that you're going to apply exactly the same way every time. All right, so if we find the smaller, then we're going to use this x is less than. Right. If we wanted to use the larger, we'd have to switch this so the x is greater than, right? Notice that it's not equal to, it's less than. All right, so we have our big S is going to be the smaller. So we had 6 pluses and 1 minus. So our 1 minus, that is our test statistic here. And so we want to find the probability that little x is less than big S. So going over to our distribution, our little x is 1. Right now we're not going to include 1 because our rule is x is less than. So we're only going to include the distribution result for the probability x equals 0. All right, now if our x were larger, if it were 2 or 3 or 4, then we would take and add up the values in the sign test table that were all less than that value. Okay. The only one here is that's less than 1 is 0, so it's p equals 0.008. Right now, at an alpha of 0.05, that p is less than our alpha, so we would reject null. Okay. So if we do want to find an exact p-value for our sign test, and we need to use the binomial table, even though we can get an accept or reject from the sign test table. All right, so let's look at the summary for our example problem. We looked at the signs and the differences. We found that we had 6 pluses and 1 minus. Using the sign test table, we knew we could reject the null hypothesis at the 0.05 level. Um, and we could draw our conclusion, which is going to be median. Remember, this is not about means. This is about medians. Median aggressiveness score is significantly different. You could say higher between female feral cats with and without kittens, right? As we might expect. And we found our exact p-value by going to the binomial distribution. The probability that x is less than 1 is 0.008. This should match the result we get with the sign test table. Okay, so one way you can do a little quality control step is to always look at the sign test table and make sure that it agrees with your p-value. Sometimes you made a mistake adding, or you did a x is less than or equal to 1, all right? And if that conflicts with your result from the sign test table, then that tells you you should look at it again, double check, make sure you did everything right. All right. The next test that we're going to look at is the non-parametric equivalent to the two independent samples. And I'm going to teach you the test by working through an example problem, um, because you sort of have to do do this sort of moving around of numbers, and it's a little different than just having an equation. All right, so we wanted to find out if taking vitamin C decreases the number of days you're sick with a cold. All right, as once you know you have the cold. All right, so we had some students with a cold take vitamin C. We had some students with a cold that did not take vitamin C, and we measured how many days they were sick. All right, so we have a pretty small sample size here. This days sick is not going to have a normal distribution, so we decided to do the rank sum test. So notice we've got 5 individual students that were in the control group. Now they were randomly assigned. And then we have the days that they were sick. So these didn't take the vitamin C. And then we have different students, right? Students 6, 7, 8, 9, 10 that did take the vitamin C. And then we have the days sick here. Okay, now it looks like the number of days sick is lower with the vitamin C, all right? The two highest measurements we have, 6 and 8 days, are in the control. All right, but we're going to test for the medians, and these much larger values are not going to have as much weight in this test. All right, so when we talk about, oh, we see if there's a difference from in our results from one group to the other, and we've we've had a lot of examples where there was a significant result. But if the variance is large enough between the individuals, then even though it looks by eyeball like the data might be significantly different, we have to do the test. All right. That's why we're doing statistics in the first place. All right, so again, our null hypothesis is that the median of the control is equal to the median of the vitamin C group, and we're going to do a not equals alternate hypothesis. And what we do to do this test, this is the rank sum test, the way we do it is we find the sum of the ranks. All right, so I'll show you how to do this, find the sum of the ranks on the next slide. All right, so in order to find the sum of the ranks, we're first going to put our data from both treatments. We're going to mix it together and put it into ascending numerical order. All right, so in this table, the first step is to put the data in order, and we're going to interleave or mix together all of the data points. And I've labeled them here by color. So the vitamin C treatments are this sort of tan or yellow, and the controls are this darker blue, gray. Right, and you can see kind of if we line the distributions of these two groups up that they're partially overlapping, right? They're not totally overlapping. Right, now we have some values that are the same in each of the two groups. We have to do something special if that's the case, right? But what we're going to do, we want to assign ranks to our ordered data. The ranks that we have to assign are as many as our total sample size. So I always start with the first draft of my ranks. All right. So this first data set, it's the smallest one. It has rank 1. This data point, number 8, is the highest one. We have 10. So we just assign the ranks 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. All right. However, we don't want to give a different rank to these two values that have exactly the same um, um, value of our variable. And we don't want to assign a rank of 3 to this number 2 from one treatment and 2 to the other treatment. All right. They should have the same rank. So what we're going to do is we're going to take these two ranks that we were supposed to assign, that we need to assign, and we're going to take the average of them and give it to both of these data points. All right. So this step of fixing the ties means that where we have two or more values in the different treatments. We don't have to do anything if they're in the same treatment. We need to take the average of the two ranks that we would assign them and then give it to both, right? So we take 2.5. Now, this is going to add up to the same total. So this adds up to 5, and these two add up to 5. And if you have more than two observations that are tied, then you average however many ranks would be assigned to those values. So we also have a tie here at the 4 level. So we average 5 and 6 and assign the rank of 5.5 to both of them. And we do the same thing here for this measurement of 5 in both groups, right? All right, so we fixed the ties by averaging the ranks. And now we have our ranks. We're going to sum the ranks, right? So we're going to sum the ranks up separately for each of the two treatments. And we're going to call the sum of the ranks test statistic capital W. So capital W for the control. I'm looking across at where these darker gray observations are. If you want to write this down on your paper using different colors of pen or circling these observations, it makes it easy to track across and know which ones belong to which treatment. Okay, so we mix the data together. Now we're going to separate it out. So we're going to take all the ranks that belong with the control treatments in the darker blue. We're going to add them up. So we've got 2.5, 2.5 over here. We're going to find the next one, 5.5. We've got it here. And then we're going to have 7.5, 9, and 10. That adds up to 34.5. Right, now we're going to also do that. So we have a W for each of our two treatments, and I'm keeping them labeled. We're going to look at where these yellowish tan boxes are. Our rank, the first one is going to be 1, and then we have a 2.5, a 4, a 5.5, and the 7.5. All right, so I've written these down over here. We're going to add up 1, 2.5, 4, 5.5, 7.5. Make sure that you are seeing where those come from. All right. So this one adds up to 20.5, and I'm going to show you on the next page how you test the hypothesis, or how you find the p-value, and we're going to use the lower, again, just like we did for the sign test. We're going to pick the lower of our two sums of ranks. So our test statistic W is going to be 20.5. All right. So how we're going to find whether we reject or accept the null hypothesis. Now, because this is kind of a really simple test, we need to use this table. I don't have a distribution that you can find the exact p-value for. We're just going to use this table. This table is set up. This is in your table packet. This is only the top left corner of it. And this table is established so that it gives us the um, critical values, W stars, for our rank sum test at an alpha of 0.05. All right, so all we have to know to evaluate our hypothesis is we to know our sample size. So remember, this is two independent samples. So we have a sample size, a little n sub 1. Right, we're going to find that across the top. And then down the side of the table, we have the sample size for our little n sub 2. Okay. And so we're just going to go across the table. And in our example, we had a sample size of 5 for both. We had a 5 in one of the treatments and also a 5 in the other treatment. So in the table, we're going to read off this number 17. All right, what is 17? It's our critical value. If we compare our W with this W star, and again, we're picking the smaller of our two treatments, values of W. If our value of capital W is less than or equal to the W star, the number from the table, then we'll reject the null hypothesis. All right, you just have to know how this table works. Right, it's a little unsatisfying, um, but it allows us to do this test pretty easily. Okay. All right, so we found that our W star for our sample sizes was 17. And let's look at the result from our test. Okay, so here's the data back up here again. And so we've got for little n sub 1 and little n sub 2 equals 5. We found our W star in the table, which is 17. Our W, our test statistic, which is the smaller of the two W's that we calculated. Now, really, if you know by looking at the data like you could see here, you don't actually have to calculate both of them. You only need to calculate the smaller one. Okay, so you don't have to show your work to calculate both, but you do have to assign all the ranks so that you can correctly find this smaller value of W. All right, we are gonna ask this question. Is our big W less than or equal to our big W star? In our case, for this example, no, it's not. 20 is not less than, 20.5 is not less than 17. All right, so we do not reject the null in this case. And you're thinking, wait, but my eyeballs tell me that it's lower. It's not lower enough that we're able to say that there's a difference, okay? So when I say these tests are not as powerful, this is what I mean. See that the numbers are all more days in the control, but it's not enough that we can reject the null. So our conclusion is going to be there is not a statistically significant difference or reduction in days sick if taking vitamin C or not. Okay, so another way we might say this is that there's not a significant effect of treating colds with vitamin C. Don't bother. I don't know. Looking at the data, maybe I would take it, but it actually turns out in real science that if you take vitamin C when you're feeling a little run down, maybe you think you might be exposed to a cold, um, like going on an airplane, it can help prevent the cold, but it doesn't actually help you once you've become sick. So that's like published papers. Okay, so we have some practice problems to do hypotheses tests, doing sign test and rank sum, and also some problems this week to help you practice making a decision of whether you need to use a non-parametric test or not, basically looking at the t-tests and seeing if their assumptions are violated. So I hope this gets you excited about a whole different style of statistical tests that can be used on a wide variety of different data types.